Hi, I'm Mark Hyde. I'm the founder of Choo-Choo's Journey, which is a fundraising and awareness campaign to benefit pediatric cancer research. I'm here today. I have the pleasure to be here with Dr. Amit Sabnis, who keeps asking me to call him Amit, but I continue to call him Dr. Sabnis. We're here today to talk to him about pediatric cancer research in general, talk about his studies and research, and we're here in his lab, as you can see, and we're going to have a chance to kind of walk through and see what actually happens in a real research setting.
So I'm an assistant professor in the Department of Pediatric Hematology and Oncology here at UCSF, and that means that I get to wear two hats. Part of that is being here in this lab, thinking about what makes cancer tick, and more importantly, thinking about how to make cancer stop ticking. And the reason that that is so important to me is because the rest of my time I'm actually over in the hospital taking care of children with cancer. And that's part of what motivates me to really try my best to push this research forward and hopefully develop new cures in the next few years.
That's fantastic, fantastic. So what is you, can you describe or what your exact research is right now that you're doing?
Absolutely. So people have been studying rhabdomyosarcoma actually for a long time, both in the clinic and in the laboratory. But I think that we're taking a slightly different approach, which I'm really excited about. So, a lot of people have studied rhabdomyosarcoma with conventional chemotherapy agents and how to improve those, how we give them, and how to improve outcomes with that. Some people have been studying the genetic changes in rhabdomyosarcoma and trying to figure out how that may help us fight this disease. But we're really interested in thinking about the proteins that make up rhabdomyosarcoma cells. And we've actually discovered that there's a group of proteins called chaperones that kind of work as safety officers in the cell floor, and that rhabdomyosarcoma cells really, really depend on those safety officers. So much so that we can actually inhibit all those protein chaperones and affect rhabdomyosarcoma cells, so make them die, but not really affect any of the other normal cells in the rest of our body.
That sounds really exciting. Can you tell me about how that approach to research and into therapy would actually is likely to impact outcomes, both immediate survival as well as long term?
That's a great question. So we know that even those patients that we can cure of rhabdomyosarcoma right now, the first time through, that cure comes at a really high cost. So there's lots of side effects from the DNA damaging agents that we use, the chemotherapy and the radiation. And the reason for that is that normal chemotherapy works by, damaging the DNA, which are the instructions for cells. And even though we think that the rhabdomyosarcoma cells and other cancer cells really depend on that way more, we know that normal cells in the body also need that DNA. And so when you damage the DNA of the cells that line your mouth and your stomach, you can get bad pain and mucositis. When you damage the cells, the DNA in your white blood cells, you can have neutropenia and risk of infections. And what we think is unique about our approach is that the chaperones that we're targeting now with these newer drugs, it seems like rhabdomyosarcoma cells need them, but other cells just don't. So even if we block these proteins or damage these proteins across the entire body, we think the side effects are going to be very, very minimal, and really all that we're going to be going after is the cancer.
Oh, wonderful. We're here, we have the good fortune to be here in the lab and to kind of see this firsthand. Can you talk us a little bit about how you take the idea and actually turn that into something which can be used across the street to help your patient?
That process happens here in the lab, and it actually is a huge multi-step process that I think you'll probably be hearing a lot about as you go forward. So, the first and foremost thing is to actually get people in the laboratory who are interested in studying and curing rhabdomyosarcoma. The fact of the matter is, as a physician, you're really primarily hired to see patients. And so for people who are trained as oncologists to actually come and spend time in the lab, they need salary support, they need protected time. And then for all of these things that line the walls, each of those reagents and different tools that we're using to conduct experiments and get answers, those can become increasingly expensive over time. And we think that that's money that's well spent, that by investing in asking the questions here in a laboratory setting, we can find the answers that we need to eventually start handing off new drugs, new therapies, new tests to our colleagues who are actually designing and carrying out the clinical trials that we'll eventually need to really improve outcomes for patients with this disease.